Hello and welcome everyone to this podcast on organelles and protein transport. Okay, so now remember, if we're talking about a bacterial cell or a eukaryotic cell, there's this fundamental problem that within these cells there are hundreds to thousands of reactions occurring, depending on the cell type and whether we're talking about bacteria or a eukaryotic cell. There are variable amounts of reactions, but there are certainly many reactions occurring at any given time. And they can't occur right next to each other or just kind of in the random mix. There has to be a strategy to make sure that when we're making DNA, that those reactions are in one area. Glycolytic enzymes, when we're going through glycolysis, those enzymes need to be in a position to function and work together. So there are two general strategies that cells will utilize. Now both bacteria and eukaryotes will use this first strategy, and that is they will aggregate enzymes that share common pathways. And we can think of as some examples protein, DNA, and RNA synthesis. Even though bacteria don't have specialized compartments, they will have regions within the cytoplasm where certain enzymes are aggregated to carry out specific functions. Maybe they're functions in translation or replication or transcription or glycolysis. And then even in eukaryotes, if we don't think about organelles yet, and we haven't talked about organelles yet, but they will also aggregate enzymes, even within an organelle. So let's look at the nucleus. In the nucleus, there are regions that are called transcription factories. And this is where enzymes that function in transcription aggregate. Okay, this second strategy is to use membrane-bound organelles that compartmentalize common cellular reactions. So if we have, say, a mitochondrion here, the enzymes that function in the citric acid cycle and oxidative phosphorylation are compartmentalized to this organelle. Enzymes in, say, the lysosome. So this was mitochondria. I'll put an M there. Enzymes in the lysosome are involved in degrading proteins and even other organelles. So you can imagine how that would be very important to keep in a membrane-bound structure away from everything else. We also have peroxisomes. We'll have the ER associated with the nucleus, and the ER will be linked near the Golgi. Each of these organelles have a very unique and special function. They contain unique enzymes that allows them to do those special functions in a confined compartment. This allows a cell to have thousands of cellular reactions occurring at the same time without interfering with other reactions. So this has been a tremendous asset to eukaryotes as they have evolved. Before I move on, I noticed on the note-taking guide I did ask to identify the endomembrane system. So the endomembrane system is a collection of several organelles that communicate with each other. The organelles in the endomembrane system include the ER, the lysosome, the Golgi, the peroxisomes, and endosomes. And we'll talk later in this chapter about how the communication of these different organelles in the endomembrane system are important in moving vesicles around between the different compartments. Now, when we look at this eukaryotic cell, we can see how it's beneficial, or at least we might presume it's beneficial, to be able to compartmentalize all these reactions. But there are some problems we should consider. The first problem is, how did they get there? How did these individual organelles arise? And then as we consider a eukaryotic cell that already has these organelles, we have problems associated with how do we get proteins into each of these organelles so that they function properly. Because remember, most of the proteins that are made in the cytosol. So they have to find some way to get to these different organelles. So let's talk about the evolutionary aspect first. Where did these organelles come from? Okay, so let's begin this discussion of organelle evolution. Where did they come from? And it's important to remember that the building of the eukaryotic cell didn't happen in just a few years or in just one round of evolution, so to speak. It happened in different stages. At various times there was an adaptive benefit to having certain kinds of organelles formed that allowed them to persist. So let's consider this ancient bacterial cell here. Probably not all that much different than a bacterial cell we would see today. It would have this plasma membrane around it. Many of the functions would have been linked to the plasma membrane. Many cellular reactions needed for survival, including citric acid cycle and glycolytic enzymes, would have been found in the cytoplasm. It would have had a chromosome, and it would have been attached to the plasma membrane. And then over time, what might have happened is in this cell here, the area where the DNA is attached, maybe it began to invaginate into the cytoplasm just a little bit, which would have pushed that DNA a little further in like so. And then over time, what might have happened was this invaginated membrane here might have fused together, generating this ancient eukaryotic cell that had the plasma membrane fused, and these areas here would have fused around each other, and that might have formed our first nucleus. Now at this point it's still very primitive, it just has a nucleus. We could also have imagined that depending on where this fusion happened, we might have still some of that membrane coming off here, like so. And this could have eventually developed into the ER, because we know that the ER is interconnected with this outer membrane of the nucleus. And we know these membranes share some common features with the nuclear membranes. So why did this occur? It could have been random, but your book poses an interesting explanation as to why a need arose to develop these internal membrane structures and to compartmentalize some of the reactions. Now remember, a bacterium cell is small in comparison to a eukaryotic cell. Because of its small size, it has a high surface to volume ratio. They can accomplish a lot of tasks within this limited size structure. But as that cell gets bigger, it's hard to accomplish those same enzymatic reactions and achieve the same energy surplus needed to survive. Now modern day eukaryotes, probably not this ancient eukaryote, but we could imagine how they got bigger and bigger over time. But the modern day eukaryote is 1,000 to 10,000 times bigger. This gives them a low surface to volume ratio. This would have made it much more difficult to survive with just the plasma membrane maintaining the structure of the cell and generating the energy needed. So the need to develop this intermembrane structures here, these compartmentalized structures, may have been a necessity because of the increasing size of the cells. So this first organelle perhaps did arise in such a way caused by the invagination of the membrane, which eventually enclosed upon itself. And then from there, reactions found in present-day ER started to compartmentalize in this extra layer of membranes. It is also likely that, in addition to the nucleus and the ER, that the lysosome, the Golgi, and endosomes, which we're not really going to talk about too much in this class, likely all arose due to invaginations of the plasma membrane. Okay, so let's continue working with this ancient eukaryotic cell and add some more to it. And let's assume that this has now also gained lysosomes, Golgi, and the endosomes. Likely at the onset of this ancient eukaryotic cell, the oxygen concentration would have been initially low. But as oxygen concentrations began to increase on Earth, there was a need for an adaptation that would allow these cells to survive in the presence of oxygen. So how was this cell going to be able to survive in the presence of this increasing oxygen environment? So how would this ancient eukaryotic cell have survived in this increasing concentration of oxygen? It turned out there were some aerobic bacterial cells that survived in the presence of oxygen and used oxygen. During an event that we refer to as Endo Symbiosis, this ancient eukaryotic cell engulfed this aerobic bacterial cell. And then what would have formed then would have been this cell here, where we still have our nucleus with the ER around it, the Golgi, the lysosomes. But now it just brought in this aerobic bacteria. And then we would have the bacterium that has just been engulfed. I draw two membranes here because we engulfed this bacterium through the plasma membrane. So we ended up creating two membranes. Now I haven't shown it, but remember that this bacterium here has its own genome associated with it, and so this one did as well. On this genome would have been all the enzymes necessary for oxidative phosphorylation. Now over time, what has happened as this new cell here, which we will call an early aerobic cell, or early aerobic eukaryotic cell, and as I was about to say, let me finish now, is the DNA from this mitochondria over time, over thousands and thousands of years, some of the genes have migrated into the nucleus. However, several genes, around 13, have remained in the mitochondria. These include genes necessary for oxidative phosphorylation, as well as genes necessary to make proteins within the matrix of the mitochondria. Now likewise, we could have had a photosynthetic bacterium that could have been engulfed by an early eukaryotic cell. And I should have left a little bit more room here, but let's go ahead and draw it in here. And again, it would also be double membrane, which allowed these cells to undergo photosynthesis. And again, I need to draw here. This would have had DNA in it, as well as the chloroplast also has DNA in it. And it has also lost genes to the nucleus over time. So with this in here, I would now call it an early aerobic and photosynthetic eukaryotic cell. Okay, now we haven't talked about the peroxisome. How did a eukaryotic cell eventually obtain a peroxisome? Well, there's some debate in the literature on how this has occurred. One idea is it formed from the plasma membrane, just like the nucleus and the ER and the Golgi and the lysosome, and that enzymes in the nucleus that were necessary for hydrogen peroxide metabolism and oxygen metabolism would have gained the ability to enter into this new structure. Another interesting idea is that the peroxisome was also involved in an endosymbiotic event. So let's talk about this proposed endosymbiotic event for peroxisome evolution. In this idea, there would have been a primitive bacterial cell that had the ability to metabolize oxygen and hydrogen peroxide, but primarily oxygen. And the idea is that this would have predated mitochondrial endosymbiosis or chloroplast endosymbiosis, at a time when the oxygen concentrations were slowly rising and becoming toxic to these early eukaryotic cells. It's possible that this early bacterial cell was engulfed by this bacterium and set up to become what we now call the peroxisome. And on those early stages, it would have been sufficient, at least the proposed idea, is that it would have been sufficient to metabolize oxygen to allow this early cell to survive. Now, the need for the mitochondria and the chloroplast came later because, or at least there was a beneficial adaptation to obtain the mitochondria and chloroplast because they provided more energy than the peroxisome. The peroxisome just protected the cells. It didn't really provide the energy that the mitochondria or chloroplast could have performed or provided. So what is some of the evidence that peroxisomes could have arose by an endosymbiotic event? So the first idea is that peroxisomes formed by the division of other peroxisomes. So you get more peroxisomes in a cell when a pre-existing peroxisome divides. This is also true for mitochondria and chloroplasts. So again, there was this connection to these two organelles where there's considerable evidence that they evolved in this manner. Another piece of evidence, and we haven't talked about this quite yet, is that they import proteins post-translationally. Proteins are made in the cytoplasm and then they are imported into the peroxisome. Just like proteins needed for the mitochondria and chloroplast function. They're folded into the cytoplasm and then taken into either the cytoplasm or the mitochondria. We'll talk in a moment, but those organelles in the and we'll see in a moment, the proteins needed in the endomembrane system, the lysosome, the ER, the Golgi, get their proteins through the ER. One idea that kind of makes us think maybe it wasn't the peroxisome is that there are no peroxisomal DNA. Now, it's possible that since the peroxisomes are believed to have formed much before the mitochondria and the chloroplast, that over time it's lost all of its genome to the nucleus. Now, the idea that supports that is that we know that the mitochondria and the chloroplast are losing DNA, losing genes to the nucleus, and they continue to do this. It's a very slow process, but they continue to lose their DNA in this manner. So this could be a precursor to what we'll see mitochondria and chloroplasts in the future. Maybe they won't have their own genomes. So mitochondria and chloroplasts and the peroxisomes are thought to evolve through the endosymbiotic events. They divide in this manner similar to bacteria. They import proteins in a similar way. And at least for the chloroplasts and the mitochondria, they have their own genome. And as I said before, the idea that they formed from the plasma membrane and ultimately through the ER is also another idea that has some evidence with it as well. Again, that's the good thing and bad thing about biology. We still don't have all the answers. Okay, let's move on and start to consider how these organelles will ultimately get the proteins they need to function.